Welcome to Veterinary. In this video, I'm going to talk about dermatophytosis. Dermatophytes are pathogenic fungi belonging to the genus Epidemophyton, Microsporum, and Trichophyton. They are the members of fungi imperfecta. That means they don't have a sexual stage, but some have shown to be having sexual stages and they are placed in Ascomycota. They have affinity for keratinized structures. That means dermatophytes are keratinophilic and they colonize and invade skin, hair, and nails. They grow slowly on specialized formulated laboratory media such as Abrotextrose agar and dermatophyte test medium. They are aerobic, just like other fungi, and the colony is often pigmented, having different color on the top and reverse side. They form macroconidia, which are the basis of differentiation of species between dermatophytes, and arthrospores, which are the infective part of this fungus. Now let us come to epidemiology. So the pathogenic genera include Epidermophyton, Microsporum, and Trichophyton. Dermatophytes occurs worldwide, so they have a worldwide distribution and they are the most important superficial mycosis in human beings and animals, especially cats. Okay, so dermatophytes are superficial mycosis, they cause superficial mycosis. Then they occur more frequently in stabled animals than those kept in open pasture. Infection by M. canis is common in cats and dogs and is often asymptomatic. So cats are the natural reservoir host of M. canis. Okay, and depending upon the reservoir, the dermatophytes are classified into three groups. Anthropophilic, they're adapted to the human beings and they include the species of the genera Epidermophyton. Zoophilic dermatophytes mainly affect animals, but they can also infect human beings. And zoophilic dermatophytes include M. canis and T. mentagrophyte. So M. canis has the reservoir host in cat, T. mentagrophyte has reservoir host in rodents, and T. verrucosum has the reservoir host as cattle. Then geophilic dermatophytes live in the soil as saprophytes, but if there is some breakage in the skin or there's some cut or if the host is immunocompromised, then the fungus from the soil can enter into the animal and cause dermatophytosis, and this will include Microsporum gypsum. Okay, so we have three categories: anthropophilic, zoophilic, and geophilic dermatophytes. Then pathogenesis. Dermatophytes invade stratum corneum, hair shaft, and claws by producing proteolytic enzymes. Okay, so what are these proteolytic enzymes? Keratinase, elastase, and collagenase. And these enzymes help them to penetrate the conified or keratinized structures. Okay, then by the process of segmentation and fragmentation, the fungal hyphae form arthrospores, and these arthrospores are the infective portion. Okay, they adhere to the keratin, they adhere strongly to the keratin and germinate within hours of contact, and then they will release the fungal products. And the fungal products, along with cytokines, will damage the keratinocytes, and there will be a cell-mediated immune response, and this will cause delayed type hypersensitivity. So the pathogenesis of dermatophytosis is based on type 4 hypersensitivity reaction. Then pathology. So there are four main lesions. First is the ringworm. Okay, so at the center of the lesion, the fungi will be dead, and on the periphery the fungi will be viable. Okay, so this will form a ring like structure, red ring, and this is called ringworm, and this lesion is very common across all species. Then we have a very important lesion in dogs, that is carrion. So they are eruptive nodular masses, so this lesion here is called a carrion. Then there are also some masses containing distorted fungal hyphae called pseudomycetoma. And there's also infection of the nail beds and nails, that is called onychomycosis. So onychomycosis is fungal infection of nails. This is also very important. And of course, scaly patches or crusty patches of alopecia are seen in all species. Then diagnosis. So the primary diagnosis for dermatophytosis is Wood's lamp test. Here, M. canis and M. distortum, they will produce metabolites like pteridine, and they will fluoresce apple green under the ultraviolet light. So Wood's lamp is a device that emits filtered ultraviolet light. Okay, and when you examine the animal under a dark room, then this light of the Wood's lamp test, it will illuminate certain metabolites. Okay, so in Pseudomonas, they will produce like blue color. In some other organism, they will produce some another color. In case of dermatophytes, they produce apple green fluorescence. Okay, then specimens. So for specimens, we have to collect the hairs and the skin scraping. So hair should be plucked and not cutted by the scissors. Okay, and any scab material, they should be obtained from the edge of the lesion. And if you are collecting the skin scraping, you have to scrape the skin with blunt edge of the scalpel until the blood is drawn. Why? Because this is the layers of the skin, this is the hair. So the dermatophyte affect this shaft and root of the hair. Okay, so basically this region under the superficial layer. So that is why we have to scrape until the blood is drawn. Okay, so we can collect the actual fungi. And if you are worried about the contamination with bacteria, then you can wipe the lesion with 70% alcohol before collection of specimen. Then we go for direct microscopy. So you take the hair specimen, you take the skin scraping specimen, and you put it in 10% KOH solution. Okay, and then after 10 to 15 minutes you observe it under the microscope. So what you will observe, that the microsporum species, okay, so species of dermatophyte belonging to the genus Microsporum, they will have thick walled spindle or boat shaped macroconidia, and a few tiered shaped macroconidia borne singly on the hyphae are seen. In case of Trichophyton, thin walled elongated cigar shaped macroconidia and many small microconidia borne singly along the hyphae or in clusters are present. Okay, so this is very important. What is the difference between microsporum and trichophyton under direct microscopy? Okay, another thing that is the point of differentiation between the various species is the number of septa and the number of cells, and also the shape of the macroconidia. Okay, so in case of Microsporum canis, you can see the macroconidia is spindle shaped. It has 13 to 15 cells and around 15 septa. Okay, so just like this, you should try to remember the number of cells, the shape of all the species of dermatophytes. Then Trichophyton verrucosum is very unique in the sense that it has chlamydospores present in chains. Okay, so chlamydospores present in chains is a feature of which dermatophyte? Trichophyton verrucosum. Then isolation and colonial appearance. So on SDA, you have to incubate the fungus aerobically at 25 degrees Celsius for 3 weeks. In case of verrucosum, you have to incubate for 5 weeks. Okay, and what you will observe that on top and on reverse side, the colors are different. Okay, so these are the colonies of M. canis, T. mentagrophyte, and T. verrucosum, respectively. Then, apart from SDA, if you want to have a differential media, then you can use dermatophyte test medium. Dermatophyte test medium is the differential media for dermatophytes, which contains phenol red indicator. Okay, and if there is growth of dermatophyte on the medium, then it will change its color from yellow, which is the original color, to red. Then we have hair perforation test. So T. mentagrophyte will invade the hair shaft and it will produce conical perforations of the hair. So these are the conical perforations of T. mentagrophyte, and on the basis of hair perforation test, we can differentiate the dermatophytes into ectotherms and endothrix. Endothrix. So endothrix will cause um, damage inside the hair. So they will perforate into the hair, while ectothrix will remain outside. Endo, perforate in the hair, ecto, outside. Then there's hair bait technique and host. They are given. So dogs and cats, they are affected by M. canis. Cattle affected by T. verrucosum. Pigs, M. nanum. Horses, T. equinum. Okay. Then fowl are affected by Microsporum gallinae. This is very important because Microsporum gallinae causes favors. What is favors? White crusty patches which develop in the comb and wattle. So dermatophytosis in case of poultry are called, or it is called as favors. Okay, so this is it for dermatophytosis. What were the important points? First, you have to remember the genus, the three genus, Epidemophyton, Microsporum, and Trichophyton. Then they belong to fungi imperfecti. They are keratinophilic. The colonies are pigmented, having different color on top and reverse side. They produce microconidia, macroconidia, and arthrospores. Okay, they are classified into anthropophilic, zoophilic, and geophilic dermatophytes. They produce proteolytic enzymes, and the main mechanism of pathogenesis is delayed type hypersensitivity. Okay, the main lesions are ringworm, alopecia is seen, carrion is especially unique in case of dogs, pseudomycetoma, and onychomycosis. Onychomycosis is often asked in the exams. Then the primary diagnosis is Woodsnam test. Okay, the dermatophytes, M. canis and distortum, will give apple green fluorescence. For specimens, you should collect hairs that are plucked and skin scraping on a black paper. Okay, for direct microscopy, we should go for 10% KOH bed preparation. There, you should see the difference between Microsporum and Trichophyton. Then, for isolation, you can do it on SDA or dermatophyte test medium, DTM. Then we have hair perforation test and hair bait technique. Hair bait technique is used to isolate dermatophyte from soil, so it is used for geophilic dermatophytes. Then the natural hosts. In this, you have to remember favus is caused by M. gallinae. So this is it for dermatophytosis. I hope you like this video. If so, please hit the like button, subscribe to my channel, and share this video with your friends. Thank you.